So having introduced the basic topic of representation and shown that it hides a number of issues, we're now going to turn to one notion, one way in which representation is discussed with respect to brains. And this way is relatively free of many of the issues that we raised. For those who are very enthused about stories involving representations and cognition, these are often held up as paradigmatic examples of representation in the brain because our knowledge here is secure. We're going to interrogate this and see what kind of representations these are. And we're going to make use of a little bit of neuroanatomy that we've already learnt. So we've looked already at the connections that run from the eyeball to the occipital lobe in the brain, which are heavily schematized in this diagram. As you know, projections of the optic nerve run from the back of the eyeball through the optic chiasma, with some fibers crossing, around to the back of the occipital lobe and primary visual cortex. Now, different nerve fibers leaving different parts of the retina run together as far as visual cortex, so that fibers which are close together on the retina project to areas which are close together in visual cortex, so that the whole spatial organization of the retina is maintained throughout several transformations. This is undoubted, and we can follow these connections. It doesn't say anything about representation. What it does say is that what's going on at the retina can be understood in relation to what's going on in the brain and vice versa, because we know the route from one to the other. Let's have a look at what happens along the way. Shown here on the top is a cartoon of the retina of a monkey, divided into three regions. The black region in the middle, we're simplifying, it's not actually in the middle, we can take to be the foveal region where the cones are located, densely packed, sensitive to color, and so on. And around that we have a perifoveal region occupied by rods, and around that again a peripheral region with less rod density. Now when we follow those three regions, divided here into quadrants, around to the back of the monkey's brain, the lower figure shows the back of the brain opened up like this. So areas which are together are now opened up. So to recreate this brain, you would have to fold those two areas back in again. And here we can see the various areas from the top reappear here in the bottom. We can distinguish the foveal, the perifoveal, and the peripheral areas. And their organization, that on top show up as the four quadrants, are preserved here. But something has also changed. Notice that the relative size of the area given over to the fovea is much larger compared with the relative size of the area of the periphery, which has got much smaller. So here we have a distortion of the kind I drew your attention to with the hand imprint in the clay. I said that the hand imprint in the clay retains its character as a record of a past event, even if we stretch and deform the clay so that some areas are either elongated, expanded, or compressed and shrunk. That kind of topological deformation is exactly what we see here. And so one could say that this looks rather more like an imprint, that is its connection, the connection between what's going on in the retina and what's going on in the brain, has more in character with the relation between the original hand in clay event and the trace left afterwards than it does like anything to do with symbols, for example. Now we find this in three sensory modalities. We find this in vision. We also find it in touch. We noted before that touch is not a single sensory modality. Nevertheless, we can trace the various receptors in the skin to where they first contact the brain, and that's in an area called the primary somatosensory cortex. Let's have a look at the top right-hand picture, the little brain there. You can see at the point where the frontal lobe meets the parietal lobe, there are two gyri, two folds. The rear one and the parietal lobe is called the primary somatosensory cortex, and that's what we're dealing with here. The one immediately forward of it is called the primary motor cortex, and we will meet that one again. So we can follow the projections from parts of the body to the primary somatosensory cortex, and there as well, like with the retina, those projections have an ordered layout. Now, the three-dimensional surface of the body is rather more complicated than the disc of the retina, and so it's not possible to map the entire outer surface of the body without cuts. And on the left, you can see for one hemisphere, you can see where the projections have come from in primary somatosensory cortex, and they make a largely ordered map. So you can see the body stretched out there. There's a disconnect as we come to the head and as we go down into the throat. But you can see by and large contiguous areas on the skin project to contiguous areas in primary somatosensory cortex. And just like with the retina and primary visual cortex, you can see that there are characteristic distortions. This isn't a big surprise, and the reason will turn out to be the same as in the visual system. So you can see, for example, that the amount of area in primary somatosensory cortex devoted to the lips is very large, whereas the amount devoted to the elbow is negligible. Now think about the sensitivity of your lips and the sensitivity of your elbow. Your lips are extremely sensitive, very, very sensitive. And your elbow isn't, frankly, unless you have a very remarkable elbow, in which case your primary somatosensory cortex will look a little different. But in general, we can see that those areas which are more sensitive on the skin receive correspondingly large amounts of primary somatosensory cortex. So these distortions are principled, and the distortions on the retina made the same kind of sense. The fovea is the area in which most discriminations are made. Very, very important in viewing, because the fovea is the area on the retina to which projections from the visual field stretch. That is, from the point at which you are directly looking at. And that's the point at which your vision is clearly in focus, you're seeing most clearly, making most discriminations. So the foveal area is very, very sensitive on the retina, like the lips. Now, as I said, this is sometimes discussed as a representation, and it is a form of representation, because we can understand what's going on in one place as being about what's going on in another place. The nature of the representation is more akin to the imprint that we saw than anything to do with symbols. We can actually reconstruct the homunculus here of the primary somatosensory cortex to view these distortions, and we get this grotesque character with very large hands and very large facial, especially lips and mouth. We'll be meeting this character again later on. So vision and touch both have this characteristic, that we understand the projections from the primary sensory surface, retina or skin, to the primary to the primary cortical areas, primary visual cortex or primary somatosensory cortex, and those projections make sense, and they involve characteristic stretchings and compressions. We find the same thing in audition, interestingly enough, and here's just a less clear diagram perhaps from a cat brain. The area circled on the left is the primary auditory cortex, which is in the temporal lobe, and opened up is there's a number of data points, and these show the sensitivities of individual neurons in primary auditory cortex for sounds at different frequencies. Now, we didn't cover hearing in great detail, unfortunately, but in your inner ear, the primary sensory surface, akin to the retina or the skin, is a structure called the basilar membrane within the cochlea. That's the curled up snail-like part of the inner ear. If you stretch out the cochlea, what you find is a long surface, and that's where the nerve energies arise as a result of sound vibration coming in. And low-frequency sounds stimulate one end, and high-frequency sounds stimulate the other end, so that the different frequencies are distinguished along the length of the cochlea. And here we can see that the different frequencies are spread out as well in primary auditory cortex. So here the low frequencies are correspond to the areas on the right, the high frequencies correspond to the areas on the left. Now, these three modalities are quite different. The surface of the body is a complicated three-dimensional shape. The retina is more or less a two-dimensional disc, and this is more or less a line. So they differ in the dimensionality, but they all have this in common. We have three maps. This is why we started with maps. These maps contain information about their respective sensory surfaces in a particular way because of these spatial projections which preserve structure. So that the structure of the stimulus on the sensory surface is preserved somewhat in the pattern of neural sensitivities. And they all three of them have characteristic distortions because we're not equally sensitive to at all parts of the retina or at all points in the skin or even at all points in the inner ear. So we get this contrast between the fovea and the periphery, or between the lips and the elbow or heels. So given what we've said, these look like maps, and maps are informative about something in very many different ways. When we say that these are cortical representations, we need to be a little bit careful, because what we can see is that the projections from the sensory surface to the cortex are principled, topological, spatial. We understand most things about them. They resemble rather more the imprint than the symbol. And I'm not suggesting they're the only two ways that we should think about representations, but here we understand so much about these that we can clearly say that yes, there's a principled relationship between the sensory surface and the cortex. There's direct mappings with corresponding distortions. What we don't have, that we have with signs, is an ecosystem of symbols set up by arbitrary convention. Instead, we can identify these translations, at least from the sensory surface to the cortex. There's a lot more going on in sensation and perception than that. So that's a really interesting case, one that we've encountered as we studied vision a little bit, and it's one of the senses that people mean when they say that there are representations in the brain. It doesn't mean that there's a mini world in the brain. In this case, it means we can follow those projections and they are principled.